Hello, everyone. Welcome to a virtual mini-symposium with Hubrecht Organoid Technology, or HUB, and STEMCELL Technologies. My name is Angela Zhang, and I'm the product manager at STEMCELL. I am your moderator for this event. If you would like to ask a question during the presentation, please use the Q&A panel or chat box on the right side of your screen, and we'll address those questions at the end of the presentations. We'll have roughly 30 minutes for all three presentations, followed by roughly 20 minutes of discussion. It is now my pleasure to introduce you to our speakers today. Our first speaker is Dr. Robert Vries. Robert received his PhD in biochemistry from the Leiden University Center, studying the molecular basis of oncogenic transformation. He continued his scientific career as a postdoctoral fellow at Stanford University. Upon his return to the Netherlands, he joined Hans Clever's group at the Hubrecht Institute studying adult stem cells. During this time, Dr. Vries was part of the team that developed the groundbreaking technology allowing the expansion of adult stem cells in vitro. The organoid technology became the basis of the company Hubrecht Organoid Technology, or HUB. Our second speaker is Dr. Sylvia Boj. Sylvia received her PhD in 2006 at the University of Barcelona, Spain, for her work at IDI-BAPS on functional genetic analysis of MODY genes in pancreatic beta cells. She subsequently joined the Hubrecht Institute on a long-term EMBO fellowship. In the laboratory of Dr. Hans Clevers, she first studied the role of TCF7L2 in regulating metabolism. Then she established an in vitro organoid model for human pancreatic cancers. In 2014, she moved to Hubrecht Organoid Technology as a group leader for the cystic fibrosis and cancer programs. In 2016, she became scientific director of HUB, with the ultimate goal of transferring scientific advances in the organoid technology to the development of new drugs. Our final speaker is Dr. Ryan Conder. Ryan is the Associate Director of Gastrointestinal Biology at STEMCELL Technologies. He obtained his PhD in molecular biology and biochemistry from Simon Fraser University, studying epithelial cell polarity in the group of Dr. Nicholas Harden, and performed his doctoral research at INBA in the group of Dr. Jürgen Noblich, identifying mechanisms of asymmetric cell division of intestinal stem cells. After spending time as a visiting scientist in the lab of Dr. Hans Clevers, he established the organoid portfolio at STEMCELL. Let me now hand you over to the speakers, starting with Rob.
Thank you very much for the introduction. We will have a number of presentations on organoids. Since I'm starting the presentations, I will give a brief introduction on what the organoid technology is, how we developed it, basically what we think it represents and why we think it is a model system that can improve the way we do fundamental research, but also the way we do drug development, drug discovery. I'll have an example that I wanted to give in this is a relatively short presentation, of course, an example that we did with the Clevers group very recently of a fundamental research part that I think is very interesting and answers a question that at least in oncology I've had for a very long time. And that is an example of how we use the organoid technology in fundamental research, after which Sylvia will continue with presenting the clinical data, so the data that actually demonstrates why the organoid model is also is clinically relevant and why it is why we call it, as you see here, a patient in the lab. So starting, the organoids are a model system that we think represents patients in a very different way. And why this is important is depicted in this slide. When we do drug development or research that goes towards drug development and understanding diseases, for a long time this whole effort has been revolving around target identification, understanding the molecular mechanisms behind the molecules and now antibodies, immuno-oncology drugs that we're designing. But the focus has often been on that side of it, whereas the models itself, the models that we use to do this research, have been traditional animal models, cell lines that we've used over the last 30, 40 years, and those models didn't change very much in that period. Nevertheless, we knew, of course, as in the bottom of this slide, that when we are doing research, medical research or fundamental research that is aimed towards understanding diseases, we are looking for something that represents patients, and we need something that represents patients in a lab environment. So this is what we aim to do with organoids, and what this presentation is about. And to introduce it, so what we did, what Hans Klevers did in his lab before, is they worked on stem cell biology. And you see here a very small detail of an intestine, and small intestine. And the important part of this slide is that when we were looking for stem cells, this is 10, 15 years ago, we knew that the intestine has a very highly dividing tissue, has stem cells in it, but what we didn't know was the identity of these stem cells. And so what Hans showed, or Nick Barker in his lab showed, that in the intestine, in this, and you can see the cells dividing here. If we look at the bottom of an intestinal crypt, then this is where the cells come from. They migrate up, differentiate, do their function, and on top of the villi, they die off and then shed off into the lumen. What Nick showed was that in this very bottom of the crypt, there are indeed stem cells. There are LGF5 positive cells that can repopulate the intestine the entire life of an animal and also of a person. And that discovery was very important. Well, first of all, because it showed us the first adult stem cell, but also because after showing this in the small intestine, the identity of these stem cells, we also saw that for all epithelium, it was actually very much the same. So we could use this identification in the small intestine to find stem cells in the colon, in the pancreas, in the liver, and the lung. So basically, in all these organs, we found in similar genetic programs and therefore similar molecular identities, stem cells of these tissues. And that is actually the basis of organoids. So organoids, as going back to the beginning, now we are aiming to develop a model system that works like an in vivo system. So what we want is to take cells out of a body, put them in a dish, and expand them without changing the cells, without changing the genetics and the function of these cells. So with the identification of an adult stem cell, what we did is we took these stem cells out of the organ, put them in a dish, and basically tried to see if we could find a method to expand these cells. And that worked, of course, and that is why we have organoids now. And you see that in this slide, when we can start from a single stem cell in the slide in the left top corner. And we can, of course, also take biopsies or resections from tissues. But to prove the point that the organoid is indeed stem cell based, we took in a single stem cell, developed the growth factor mix that is required for this in the extracellular matrix, and that actually gave these cultures that you see, and we now call organoids, that are on the one hand contain stem cells, and stem cells that are genetically stable. So you can grow these, you can grow them for a year, you can grow them for 5 years, and the genetics remain stable. But, and that was an interesting finding and a little bit of a surprise to us as well, that we did not only expand these stem cells and just made a dish full of stem cells, the cells orientated itself into a morphological structure that was reminiscent of a normal epithelial structure. And then, so what an organoid really is, and it's, as in the picture here, it's a little bit of a sort of a fumbled up ball here, but nevertheless, it is a structure where the basal membrane of the normal epithelium is on the outside, and the luminal part is on the inside. It has stem cells, it has differentiated cells, and basically functionally also represents a normal epithelium. So what we all of a sudden have is a model that we can take from intestine in this example, but also from the different tissues that I just mentioned, as long as it's epithelium, that when we expand it in vitro, we can make a virtually unlimited amount of cells, but the genetics of these cells remain as in the patient. Also, a large part of the phenotype, so the epigenetic marks that were on the samples in the patient remain the same. So all of a sudden we have from all these different organs, and what we know now we can take it from all these different patients, so it doesn't work to grow these organoids from 1 out of 1,000 patients, but virtually from every patient that we take biopsies from. So we have all of a sudden a way to expand these cells as if they were HeLa cells, but with the genetic and phenotypic and functional characteristics of a normal epithelium. And that basically is what is where an organoid is a different model system? What is the major change that this technology can do? That by allowing you to grow in vitro cells that you can use for normal molecular biological techniques like CRISPR, as is indicated here, but the techniques that we have been used to for the last 30 years in the lab, but nevertheless have the results be representing as if it was a patient. But Sylvia will get back to this patient representation of these cells. I will show one example, which I think is very interesting, that was published a couple of months ago, just to show you how you can use this system, and because of this patient-like, to do fundamental research questions as well as these clinical questions. And it is based on intestinal oncology, and at least when I was studying, and we've known for a long time that the bacteria in the gut, they play a very important role. They have a positive role, but they also can have a cancerous role, and a tumorigenic role. So, and for a long time, we thought that bacteria can actually cause or make people susceptible to tumors. But Hans did is look if that is true. So he used, as you see here, the isolated E. coli from different patients, and one of them was an E. coli that expresses this operon peak with the specific molecule colibactin. So it's a molecule that is expressed in this from this PKS locus that is depicted here. He also made an E. coli that has this locus deleted. And what was known from colibactin is that it is a mutagen. So actually, for a long time, people thought, what is it? Does it cause something? But that E. coli expressed bacteria in the intestine that potentially can be mutagenic was already known. So what the people in Hans' lab did is they made organoids, and they continued to inject these E. coli into these organoids. It would then treat them again with antibiotics to remove it, re-inject these organoids with E. coli, and they did this for up to 5 months. And to basically see, does this mutagenic molecule cause mutations in an epithelium that might be causing cancer. After this 5 months, they took the organoid, they made single cells, grew out organoids again from these single cells to get clonal lines and sequence them all to see in this one that expresses this mutagen and the one that doesn't express the mutagen, do we see a genetic signature that might be indicative of actually one of these of an oncogenic mutation. And when you see here the pattern in the paper, it has the whole signature. It turned out to be a signature that they find when they subtract the signature that they see in the E. coli and the control E. coli and the mutagenic E. coli. Signature that was found previously in cells. So you see it here again, just very briefly. So indeed, we have the scheme. So we infected these organoids with the different E. coli. You can see here in the slide that indeed an organoid full of these bacteria. And then with these, after 5 months sequencing these single clones of these organoids, what they found is a very specific mutational signature. When comparing, and that you see here on the left, to signatures found in tumors, actually was something that was already identified in colon cancer. So you see that here in the column of the colon, that mutational signature identified here was the same as what had been identified actually by Stratton also recently in colon cancer. And then when looking at mechanistically, they saw that indeed in APC, that's one of the causative mutations in colon, and in unstable tumors in the bottom, MSH, these genes contain mutations that cause cancer, and these mutations are caused specifically by this introduction of these bacteria. So this was, and this is why I showed it, I think it's a very interesting example that this fundamental point that we've had for a very long time, how do bacteria cause potentially cancer? We know that there was a link, but how does it do that? When using these organoids from patients, we can directly see this. We can do a fundamental question. We can see a mechanism that identifies indeed E. coli as a causative agent for cancer. And we can compare it to the patients that they come from and see that indeed this signature is present in normal cancer patients. So in summary, This is an example, and here you see a list of all the different things that are possible with organoids and that we're currently doing at Hub. The fundamental question, the fundamental research part is one of these things we do at our biobanks. And basically, it summarizes in this slide that what it means is that we can take, on the one hand, very fundamental questions in this diagram and use patient-derived material in the lab to research those. We can also use these findings to link them directly back to the clinic because we are in fact working with patient material. And the other way around, as you see in the end of the E. coli study, we can take clinical samples and see whether they indeed have these phenotypes that we find and then do the presented mechanism and look back at those samples into a clinical application. So that's what I wanted to show as an example of this fundamental research that we do with organoids. And then Sylvia will go further into the presentation of organoids in a clinical comparison. Thank you very much.
Thank you very much, Rob, for this presentation. We'll now continue with Sylvia.
Thank you for having me. I'll now talk about the predictive clinical response that Hub organoids show. And of course, we always like this slide because it brings the important message for us that with this technology, with these organoids that Rob has explained in the previous presentation, we are able to bring patients back to the lab. So we are providing a very relevant clinical model for the preclinical phases in terms of drug development and drug discovery. So actually at Hub, we've spent some effort to generate very valuable biobanks where in case of human organoids, we can represent multiple tissues like breast, colon, kidney, but from multiple diseases. Of course, oncology is a very relevant model, but we also have organoid models for genetic diseases like cystic fibrosis, or more complex diseases like inflammatory bowel disease, or also organoids, and especially nowadays became quite interesting. Organoids can be used to study some infectious diseases. And what is very important for us is reflected in this table where you can see that being organoids a very clinical relevant model, and the most comparable model could be the patient-derived xenotransplant mouse models. Organoids have all the benefits of working with an in vitro cell culture. They have a high scalability. They have a very easy implementation in the lab. Very reproducible results. You can work at throughput level, and compare with dysphagic models, the cost is quite affordable. So I'll just dedicate the next few minutes to show you a very important aspect of these organoids, apart from all the characteristics that Rob has mentioned before, and it's about their clinical validation. And this is just in these images, you can see an example of a patient with a colorectal metastasis in the liver, which tumor was observed, and a biopsy was collected in order to generate organoids. And this is the picture in the pre-treatment section. Then 2 months later, the progression of this patient after receiving some treatment was monitored. And as you can see in the post-treatment, the lesion increased in size, indicating that the patient was not responding to the treatment. And actually, the organoids of this patient that were generated in the lab and in an in vitro test exposed to the same treatment also showed the same response, meaning that they were resistant to the action of this drug. So this is an example of how these cancer organoids can predict the response of the patients. And here in the right, we try to summarize results that has been generated from different labs, independent labs, showing the same trend and the same response. So also from a point of view of the technology, the observation that different labs implementing this technology obtain the same results is also a very strong and robust message. And we have examples not only in colorectal lesions, but also for primary breast cancers. This is another example that was published in a publication from our academic partner, the lab of Hans Klevers, where organoids were generated from different breast patients, and for 2 of them we had information about the patient response. So One patient, in this case called patient 1, was identified as a tumor responder, while patient 2, the diagnostic was progressive disease, meaning that this patient was not responding to the treatment. And when the organoids of these 2 patients were generated and exposed to the same drug that the patient was receiving in a screening plate, we could observe that indeed the organoids from the patient that had good response, labeled in green, were more sensitive to the effect of the drug compared with the organoids from the patient that had which disease was progressing and there was not clinical response. So these are very encouraging results that provide extra value to the relevance of using this, in this case of oncology, these tumor organoids for drug development and drug resistance. The fact that we can have a patient relevant model in our bench to develop new drugs. But it's not only in cancer where organoids have a very big impact in terms of clinical relevance and also for applications and diagnostics. And another area where we've developed a lot of progress with organoids and where organoids are having a huge impact in the treatment of patients is cystic fibrosis. For those that are not familiar, cystic fibrosis is a genetic disease. It's caused by mutations in the CFTR transporter. Here you have an image of this transporter. So this molecule is responsible to the transport of chloride into the outside the cells. And actually there are multiple mutations that can impact at different levels the functionality of this transporter. So they've been identified worldwide as almost 7,000 different mutations. And an important aspect is that only 10 mutations are represented in 50% of the population. So in this disease, we have a big number of patients with a specific mutation. The number of the patients that has this mutation is relatively low. So CFTR is a protein expressed not only in the lung, that is the tissue that we know has an impact on the span life of the patients, but it's also expressed in the intestinal epithelium. So together with our collaborators in UMCU Utrecht, in the Wilhelmina Children's Hospital, we developed a screening assay that is what you are seeing here in these images, the FIS assay, where rectal organoids were generated from these patients. And we are able to measure the capacity of these organoids to swell. And this is the image you have on the right, when the organoids from patients with cystic fibrosis are responding to the treatment. So the image you have on the left, the organoids are not receiving any treatment, but despite there is a stimulus to induce the swelling, nothing is happening in these organoids. But if the organoids, as is shown on the right, are exposed to the treatment, and then we do the stimulus, then you can see organoid swelling. And if we are considering to use this test to evaluate restoration of CFTR, an important aspect that we are using rectal organoids and not lung organoids, for example, is that all the swelling capacity we see on the rectal organoids is 100% dependent on CFTR activity. It means that we have a very clean in vitro readout for compounds that can restore CFTR activity. So if we want to move this assay with organoids toward diagnostic, an important aspect is the reproducibility of this assay. And this is just an example of an organoid culture from a patient with cystic fibrosis from which we run the FIS assay. In this case, this is the outcome of analyzing the images you've seen on the slide before, where we can measure the swelling of these organoids. So in case of this patient, when the organoids the swelling is measured over an hour, and they are exposed to a combination of 2 drugs, the drug 770 and 809, you can see an increase of almost 180% of the area of the organoids. And these are 3 independent experiments happening in 3 consecutive weeks. So week 10, week 11, and week 12 after the establishment of the organoid culture, showing the reproducibility of the results. And in the graph below on the right, you can see the area under the curve of the graphs that you are seeing here. But very importantly, these organoids were frozen, so we can store them in our fridge. But then if we thaw them again and put them in culture, and in this case, these organoids were passaged weekly for 28 weeks or 58 weeks, so this is more than a year, and we performed the same assay. You can see that we were detecting the same sensitivity to the compound. So this brings a big opportunity for these patients, because we can generate organoids from these patients. We can freeze them and put them in culture anytime we need to perform a test with being confident that the organoids are not losing their capacity to react to the expected drugs. And of course, in the same way that I was showing before, how tumor organoids can predict clinical response, we also have shown, in collaboration again with our collaborators in the academic lab in the Children's Hospital in Utrecht, we have proven the predictive value of the organoids towards the response of, in this case, patients with cystic fibrosis carrying a specific mutation, S1251N, to different treatments. So again, this is the layout of the same experiment. Is based on the FIS assay that we expose the organoids to different compounds. In this case, is the compound 770 together with other available treatments for these patients. And then in this case, what we did was to compare one-to-one the organoid data with the clinical parameter that measures, that evaluates the impact of the treatment to these drugs. So in case of cystic fibrosis, the most relevant clinical parameters is FEV1, the capacity of exhale air volume on a second. So the higher is this value, the better lung function the patient has. And the other parameter is the sweat chloride, that it's the readout where it can measure the amount of chloride ions that you can have on your sweat. And what is very important, and it's reflected on panel K, the correlation already between the two clinical parameters is not necessarily very good. Here we have with an R of 0.2, meaning that indeed, based on clinical parameters and the combination of them, sometimes it's very difficult to see a correlation. But what is very important is that in terms of the organoids showing correlation with the clinical data, we have a much higher R, so very close to 0.5 for FEV1 or 0.8, 0.7 to sweat chloride, so that when we do the ROC analysis to evaluate the positive predictive value and the negative predictive value, in this case, when we compare organoid data with clinical data, we have very promising results with, in this case, 100% of positive predictability and almost 80% of negative predictability. So actually, with this data, already in the Netherlands, there are already patients with cystic fibrosis that they have get access to treatment thanks to the organoid response, and especially has been patients that, because of their genetic profile, they will have not had necessarily easy access to compounds. So thanks to organoids, we've been able to improve the health of cystic fibrosis patients. And together with the data that I've shown about the oncology organoids, we are expecting to have also impact in the treatment of patients with some specific cancers. And with this, I'll finalize my presentation, and I thank you all for being here, joining our symposium, and ready to answer any question. Thank you.
Thank you very much, Sylvia, for this presentation. Let me now hand you over to Ryan.
Hi, as mentioned earlier, I'm Ryan Conder, the Associate Director of Gastrointestinal Biology at STEMCELL Technologies. And today I'm excited to join Rob Vries and Sylvia Boj of the Hub, discussing the functional applications of organoid cultures. What I'd specifically like to convey through this talk is how organoids, or the organoid technology, has been able to adapt to the unmet needs of researchers and advance the science. So organoid history began about over 10 years ago now, and passageable organoids were first published in the lab of Hans Clevers by Toshiro Sato. And what he was able to do is take single LGR5 positive stem cells, isolate these, combine them with a medium which was designed to replicate the intestinal stem cell niche, add in a basement membrane extract in the form of matrigel, and 13 days later, see the structure seen here, which represents the intestinal epithelium. This was called an organoid, which was defined to both have the physiology and functionality of the starting tissue. Since then, many other organoid models have been developed from various tissues, both from primary or adult stem cell derived biopsies, as well as pluripotent stem cells or induced pluripotent stem cells, where the stem cells are driven down at one of the 3 germ layers and then further differentiated into the tissue of interest. This image here comes from a review article published by Madeline Lancaster and Jürgen Knoblich, and they speculated key areas that would benefit from the organoid technology, these being disease modeling, drug efficacy testing, drug safety testing, and organoid replacement therapy. Many papers since then have demonstrated great advances within these areas, and I'd like to highlight a few. Joanna Deckers at Jeffrey Beekman's group was able to develop an intestinal organoid model to detect a functional CFTR protein. Now, defects in the CFTR protein are what conveys the disease cystic fibrosis. What they were able to do is, upon the addition of the drug forskolin, mimic fluid uptake in the form of swelling, and within 30 minutes, come up with an in vitro diagnostic that could classify either a normal or non-functional CFTR protein. This has further been incorporated into a diagnostic now used in the Netherlands and throughout Europe. Drug efficacy and safety testing usually come in the form of screening drug libraries on biobanks, which are now very prominent throughout the world. The first of these biobanks was published in the Clevers group by Mark van de Wettering, where he took 20 samples, both normal and tumor, from patients, characterized these organoids explicitly, and then are able to use that exact library on various drug screens. This original library is what makes up the biobank at the hub, although it's expanded vastly beyond 20 patients right now. Finally, organ replacement therapy is just coming into practice at the moment, and a very eloquent experiment which demonstrated this was a collaboration between the Clevers and Watanabe groups. In this experiment, labeled wild-type organoids were taken from one transgenic mouse and introduced into a mouse that's a model for inflammatory bowel disease. So this recipient mouse has a severely damaged intestine, and as you can see from this image in the bottom left, the labeled organoids incorporate perfectly into the epithelial monolayer, hijacking the endogenous stem cell niche and therefore performing the functional aspects of the intestine. So this functions as a proof of principle for organ replacement therapy. And what I'd like to highlight from these publications and the many others that were in these various areas are organoids. The organoid technology really has become so prominent because it has advanced with the needs of the researchers. And over the next few slides, I'd like to show more current examples of how these organoids are being used. So an area that's very topical right now is infectious disease modeling, both in bacteria and this virus image we've all been too accustomed to seeing lately. Bacterial injections were first demonstrated by Sina Bartfeld in the lab of Hans Clevers. She was able to manually inject labeled H. pylori into gastric organoids. And you can see from the immunofluorescence image on the right, bacteria surviving quite nicely within the lumen of the gastric organoids, but only in the injected organoids. The non-injected organoid below gets no contamination. So this was a very nice model, but obviously has its limitations in terms of throughput. This was alleviated quite recently from publications from various labs, including Thaddeus Stappenbach and Bruce Valence, where they were able to take organoids derived from patient biopsies, further dissociate these into monolayers in a transwell system. And what this does is it effectively opens up the organoid and allows researchers access to both the apical or luminal part of the intestine, as well as the external or basolateral through introduction into the medium. A very nice example of how this is used came from a publication of Noel et al. In the lab of Nick Zachos at Johns Hopkins University, where he was able to take labeled E. coli cultured along the apical surface of the intestinal organoid layers, and then introduce a labeled macrophage into the basal medium. From the immunofluorescence image below, you can see the macrophage labeled in red has migrated through the epithelium and engulfed the labeled E. coli, as pointed out by the yellow staining at the arrowhead. So this demonstrates both a co-culture system as well as using an organoid system to study immune function. Some more current research, specifically on SARS-CoV-2, come from publications just in the last weeks. A few of them, one by Faisal Elstone and my group, demonstrates the receptor necessary for viral replication, ACE2, is expressed along the apical surface of the intestinal organoids. Another publication from Lemers et al. And Hans Clevers group demonstrates functionality. And if you look at the electron-micron graphs here, you can see that within the middle square of panel A, blown up in figure D here, you can see the virus accumulating along the apical surface and along the brush borders of the intestinal epithelium. You also see virus surviving in the lumen, and you see dead cells extruding from the epithelium, confirming a viral epithelial interaction. Another publication from Zhao et al. In the Yuan lab in China demonstrates the functional infection, as seen in green, by using antibodies against the nuclear capsid protein, and also demonstrates functionality looking 48 hours after inoculation and seeing a significant increase in viral load, demonstrating the functionality within the organoid cultures. So these data combined, as well as the publications, really demonstrate how the intestinal organoid models is an excellent one for studying SARS-CoV-2 infection. Now moving to modeling other tissues, specifically the kidney. Many labs a couple of years ago, including that of Josip von Ventra and Melissa Little, were able to come up with protocols to take human pluripotent stem cells and drive them down the kidney developmental pathway. So in this protocol highlighted by Benjamin Friedman, he's able to take human PSCs, overlay them with matrigel, induce differentiation to the late primitive streak, and then finally pick the tubular kidney organoids 18 days later. These are very well characterized within the publication, and as you can see, the various components of the kidney, the podocytes, as shown stained by the PODXL protein in magenta, the proximal tubules, demonstrated by staining of the LTL protein in green, combined with the CD31 positive endothelial cells in the system, which is a natural co-culture arising from the PSC cells. This system also works very well for disease modeling. Polycystic kidney disease is a disease that manifests as fluid-filled cysts arising in one's kidney. Through the gene-edited modification of the PKG gene, you can see, or D gene, you can see that the organoids represent a very, very, very clear phenotype representing the fluid-filled cysts, thereby enabling this system very useful for study of such diseases. Kidney organoids could also be used in SARS-CoV-2 research, as demonstrated from this recent publication from Schutkens et al. In Nature Biotechnology. Now this takes a primary derived kidney organoid, both possible from taking kidney biopsies, as well as stem cells extruded in urine samples. So they were able to characterize this through the protein markers as seen through the immunofluorescent images, as well as infection of BK virus, demonstrating the functional aspects of this kidney organoid model. SARS-CoV-2 comes into play in another publication, which was a collaboration between STEMCELL Technologies and the labs of Josef Penninger, Ali Mirazimi, and Nuria Monserrate, where they were able to take PSC-derived kidney organoids, infect with SARS-CoV-2, demonstrate an increase in viral load, as seen as the graph on the right, and then also look at a treatment through the recombinant soluble ACE2 protein. And you can see a distinct dose-dependent response when treated by this molecule. So this is now in phase 2 trials within Austria and Denmark, and hopefully will lead to possibility of treatment of the SARS-CoV-2 pandemic. There are also clinical uses of organoids. One example of this comes from microfluidic chips. A couple companies have been leading the field in this, these being Emulate and Momentus. Both have slightly different chips, but both publish quite readily on the concept of using organoid models as the epithelium within this chip. And these data, recently published by Cassandra et al. In eLife, demonstrate a distinct comparison between using immortalized cell lines, in this case the CACO2 intestinal line, compared with duodenum-derived intestinal organoids. And you can see from the differences between efflux and uptake transporters, as well as metabolites, such as the cytochrome P450-3A4 expression, there's a very clear difference between the organoid culture and the immortalized cell lines. Therefore, to gain the most physiological relevance, intestinal organoids or other types of organoids in chips should be used. Returning to the transplantations, a couple of recent publications highlight the possibility of this. In one, Giovanni Giobbi in the lab of Polo De Copi, as part of the ERC Intense grant, which both STEMCELL Technologies and the Hub are part of, demonstrated the development of an ECM derived from a decellularized extracellular matrix. Now, what this enables is the possible transplant into patients, and this was done in proof of principle by taking pancreatic organoids and transplanting within a silicone ring into immunodeficient mice. This was characterized at 2 and 8 weeks to demonstrate the functionality after. Another transplant paper came from the lab of Michael Helmrath in collaboration with Jim Wells at Cincinnati Children's Hospital. And this time they took pluripotent-derived human intestinal organoids, and in a very unique experiment, were able to take the mouse mesentery and use it as a vasculature for the human intestinal organoids. And what they found is when these were transplanted, they were able to develop both in maturity and functionality, having the vasculature. So what these studies convey is a quick movement towards future applications of organoid cultures. So STEMCELL Technologies is very active in supporting all of this research. We have 2 new products coming out and one that is just released. The ones coming out are the intesticalled organoid differentiation medium. And what this does is it expands on the current intesticalled media, which supports the stem cell expansion. This now specifically differentiates into enterocyte, goblet, and enteroendocrine cells, allowing experimental conditions under more physiological-like conditions. We have a serum-free intesticult, which is coming up, and this one now is able to remove the conditioned media component of the medium. And you can see by the bright field images on the right, over 4 different patients and 10 different passages, the reproducibility of this medium. So by removing the conditioned aspect, you get something that can definitely be more translatable. And finally, the STEMDiff kidney differentiation kit, which takes human pluripotent stem cells and drives them down the kidney differentiation pathway. And you can see from the immunofluorescence represents the key cell types of the kidney. So with that, I'd like to thank you for your time and be happy to take any questions.
Thank you very much, Rob, Sylvia, and Ryan for that excellent presentation. Okay, so we have a mix of very interesting big picture questions, conceptual questions, and very technical questions. So let me start with the first question, and this is actually directed to Dr. Robert Vries. And this, I think one of our attendees submitted this question, sort of adding more details to it throughout the presentation. So what are the top 3 applications of organoid technology in the near future, and especially in the context of development of effective drugs? I know you guys already talked a bit about that, but can you pick your top 3 that you see the most sort of future sort of benefits for?
Yeah, thanks, Angela. I think that if we look at the things that we're currently doing, using organoid technology in a preclinical phase for drug development, that now, and I also think in the near future, will be the biggest expansion in the use of organoid technology. Basically, adding new organs, the co-culture system with the immune system allows us to address a whole range of diseases we couldn't do before, and do this preclinical drug development more efficiently. That being said, what I think is the interesting part, as was in Sylvia's presentation, that it seems to be so correlative to the patient response in the clinic that we want to use it in the sort of immediate future for a diagnostic testing as well. So it seems that it's starting with cystic fibrosis, but also in oncology, the organoids mimic this patient response so precisely that we can really start to do actual personalized medicine with it. So that, and I would say, then is the sort of the number 2 or the next step. And then finally, and this is where I think this preclinical and clinical work really nicely comes together. The big thing in drug development, of course, or the problem is these failures that we have in clinical trials. And because organoids allow you to have a representative sample of patients in a biobank, a group of patients, you can basically stratify patients much better, we think, than we could before, and do a bit of this clinical trial phase already in a preclinical stage. So by making these biobanks, by generating this resource, we can, in a way, let's say, take a little bit of the clinical phase, add it to the preclinical phase, and have a much more efficient and faster drug development. So that, I would say, is the more interesting long-term approach for us, and the way we help try to develop the system.
Thank you so much, Rob. And I think that actually addressed several questions that came up along those veins. So to the attendees, if you have additional questions, in addition to what Rob has already addressed, please submit those. Let's go to the next question, and I would like to direct this to Ryan. So if you could just unmute yourself in anticipation. The question is about how are we going to make organoids an accepted model, considering the variability from batch to batch, and this came in during the initial sign up.
Yeah, thanks, Angela. It's yeah, a good question. I guess I'd like to focus on how are we going to make organoids an accepted model. I think the we is pretty important here, and we would be the entire community. And I think the way to do it is really keep pushing the boundaries and getting these publications, but also to understand what the limitations are. For example, looking at differences between the adult-derived stem cells and the PSC-derived, you get different levels in maturity, and importantly, you get strictly an isolation of the epithelium in the adult stem cells, which is important for different questions. As far as variability, I think we've worked extremely hard to limit variability, and I think using media that we know has very distinct performance criteria batch to batch, this will limit this as much as possible. We do understand there's questions that you want to access and want to be able to control exactly how you're going to modify the organoid cultures, but I think these can be used in coordination with a standardized medium.
Thank you so much, Ryan. I think this really speaks to what Rob mentioned earlier about, bringing this media or this culture system to be representative of patients. So that reduced variability is obviously a very important point. So let's let's address a question back to Sylvia. Actually, there was a number of questions around the access to organoids. So the question is, what is the best way for academic groups to obtain access to organoids for their research purposes? And additionally, there was a later question specifically around, I believe this is canine organoids. So could you address both questions, please?
So regarding the first question and what is how to academic groups can obtain access to organoids, we've been working because we realize that it's a very important model to implement in the academic background. So we have made a big initiative with ATCC together with Sanger Institute and the NCI Institute to make available organoid models from different cancers, different type of cancers, and this is a very the easiest and a very way to get a very good collection of organoids because they are fully characterized. We also are aware that, for example, STEMCELL offers some murine models organoids, so this is also a very important source. And then, of course, we also have some biobanks, an internal biobank where from cystic fibrosis, organoid models and other monogenetic diseases are also available for the community. And of course, as a last result, if these academic groups can have interactions with hospitals to get their own tissue through publications and other sources, there are already protocols of how these models can be established from original tissue. And then your second question was about canine models. At least at Hub, we've developed a model for toxicology. So this technology that was started in mouse models and then, of course, immediately moved to human is also applicable for any mammal. So we've generated, in this case, organoid models from different strains of dogs, and our main application right now is toxicology. But if there is any animal model, any dog animal model with disease from different tissues, it will be possible to generate organoid models from this animal model.
Thank you so much, Sylvia. So let's switch gears a little bit. There's a number of technical questions. And again, there's a few sort of along the same theme. So this question is related to the specific application of gut organoids to study the interaction of bacteria with gut epithelial cells, including M cells. The comment is, it would be much easier to grow open organoids for the experiments instead of microinjection. Which you guys have already talked about in your presentations. So the comment is, are there any protocols available? And then there's other questions around using transwells for this purpose. So I will direct this question to Ryan. If you could please unmute yourself and see if you could help our audience here.
Yeah, thanks, Angela. Getting into the monolayers, I think it's a really nice advancement for the field. I showed a few examples of the bacteria co-culture there, but with our with both the IntestaCult product and the new differentiation medium that is coming out, we have various protocols, troubleshooting, comparisons between different patient samples. And cell lines. All of this is available on the STEMCELL website, and we also have tech support to talk you through it. In principle, it's an extremely easy model. You just associate the actual 3D organoid models, and that part I just want to comment is extremely key. This technology, which existed in immortalized cell lines, monolayers have always been around and been responsible for a lot of the drug screening that's gone on. The organoid technology is really what enables this, because going straight from biopsy, it's much more difficult to establish these monolayers. So there's a really important scale-up step necessary in the three-dimensional organoids. So with a combination of those, the monolayers are effective. The one part of the question, specifically looking at M cells and immune function, there are a few publications on how to drive these. I think it's mostly rank ligand and NF-kappa B signaling. But these I think are still need to be perfected. And the early immune studies are coming, as Rob alluded to, and are excellent. But I think this is a field that needs to develop a little bit.
Excellent. Thank you so much, Ryan. Let's go sort of to a more conceptual question, and I'll direct this question to Sylvia. And this came in during the initial sign-up. So how to develop circulating tumor cell line? Is there any chance organoid culture can help to develop CTC cell line?
So we believe that it's possible to generate these organoids from these cancer circulating tumor cells. It's important to understand that there is, of course, the necessity that these cells have the capacity to proliferate, but it won't be different from the possibility that we have already proven that organoids can be generated from other fluids like urine or some nasal wash of the epithelium. So we don't see any problem on the possibility to develop organoids from this model.
Great, thank you so much, Sylvia. Okay, so how about a question to Rob? And this came in during the initial sign-up. So how do we solve the absence of vascularity, I guess, vascularization in organoid research?
Yeah, that's an interesting point. I think we get that question from time to time. I think the first thing is whether it is an issue that it's absent, because at the initial point of the technology was to have a purely epithelial culture, to be able to study the effects on the epithelium only without what we see in the old-fashioned, let's say, tissue culture system, where we have a mix of all sorts of cell types and tissue types together, and therefore it's not very clear what the individual cell types are actually doing. So that was the reason to make it an epithelial only system. From our perspective, we figured, so what's the next step to do? And we figured, as Ryan already said, infectious diseases was the next step, but also the immune system. Currently, we're working on systems with other stromal components. The mimetas was already in your presentation, I think Ryan's presentation. They are working with us and in this combination with STEMCELL as well, to develop a vascularized system to basically mimic also transport out of the blood system into the tissues. So as a conceptual point, I think the strong point of organoids are to have a patient-like system that is very specific to the individual parts of tissues, and then to sort of add modular components to it to add and ask all these individual questions of a more complex system.
Thank you so much, Rob. Okay, let's go to another technical question. And there's a number of questions on this. So this is something I don't believe we talked about too much today. But the question is, I am interested in liver organoids, but it's not much is known about it. What are the signature characteristics, markers of liver organoids, and how can I check whether the clumps at which have liver organoids by the simplest way? So it's really more around just liver organoids in general. I will direct this question to Ryan to start us off. If you could unmute yourself, perhaps speak in general terms if you would like.
Sure. Yeah, we didn't touch on liver organoids at all. Strange. I think Sylvia was on the original liver paper as well. But I guess Rob, when he was speaking of the origins of organoids and really focusing on the intestine, starting there because it's such a highly proliferative model, I think there was always the assumption that there was this super proliferative stem cell, and as noted when Nick found it, it turned out to be exactly as expected. This super highly proliferative stem cell that's constantly repopulating the intestine. I think the liver in principle is a very different tissue, and the organoid technology has really led to advances in this, demonstrating that there's an enormous amount of plasticity within this organ, and it's upon damage and other signals that proliferation occurs. So the liver model's been a little bit different, and there are some publications demonstrating on how to use it. We have media in development, which is expected to be out within the next 6 months or so, which really focuses on the expansion of the hepatoblast, the liver progenitor, and then has a further step to differentiate it. And I think this one, this whole system will really benefit the field, getting into the drug screening and efficacy testing that Rob was talking about, being able to detect toxicity in the term of metabolites and key functions in the original drug screening tests, way before it gets into patients and phase 3 samples. So I'm pretty optimistic of what's going to happen with the liver field here.
Thank you so much, Ryan. So we'll just go for another 10 minutes just for the audience. We do have a lot of questions, so let's try to see if we can get perhaps 2 or 3 in. So have been quite a few questions about lung organoids. Again, this is something obviously both Hub and STEMCELL have some experience with, but we didn't actually have time to touch upon today. So a number of questions about, have you studied SARS-CoV-2 infection in lung organoids, or have you cultured lung or airway organoids? I will direct this to Sylvia or Rob to see if you guys want to talk about the work you guys have done on this.
So I'll take the question, and indeed the group of Hans Clovers have a publication where it was shown how airway organoids can be generated from resections and biopsies, but also from this nasal lavage. And in this publication, and also with some characterization that we've done at Hub, we can see that in this organoids of the high airway, we represent the different cell types. We see the pseudostratify epithelium with the club cells and the cladder cells on the same disposition that they have in the tissue of the patient, and they are suitable for multiple different type of assays, including also the same type of monolayer assay if you want to study some barrier function. And we also have generated organoids from cystic fibrosis patients, and also see their possibility of swelling compared with the rectal organoids, and also, of course, as a proof of concept, generate organoids from non-small cell lung cancers. So again, with this technology and in terms of lung, we have a system that allows to generate different disease model for this specific tissue.
Great, thank you so much. Ryan, do you want to just add on a little bit more from our side before we maybe move on to the last 2 questions?
Yeah, you can point out or highlight some of the publications, or the SARS-CoV-2 publications, especially. There are other lung models looking mostly in air-liquid interface, which complement the 3D models Sylvia's talking about in the swelling assays. There is a lung organoid model come, and as Sylvia had mentioned, we are working with the Hub to develop these. So yeah, the lung field, especially what's being highlighted right now through the viral research, I think we'll be able to work very well in the future.
Thank you so much, Ryan. Again, there's a lot of questions we won't get a chance to get to, but we will do our best to get back to you on those. So how about we just end with a big picture question, and I hope to hear from all 3 speakers today. And this is a very sort of blue sky question. Where do you see challenges and opportunities for organoid research in the future? Why don't we start with Rob?
Yeah, I guess that's a good point to end with. The organoid point, and as just to add a little bit to what Ryan and Sylvia were just saying, we see for the adult stem cell cultures, we do it, but also Ryan described the IPS cultures, that what it did and what it added to the field is that we now have patient samples. So we can really do the research in the lab, not on a HeLa cell or whatever cells we've always been using in the past, but we can generate samples that are like organs and function like organs. And in fact, in the case of adult stem cells, are responding like the patients are doing. So that conceptual change, I think, is whether it's liver or lung. And the surprising thing since the first time we make an organoid was that now we understand it, we can do it from all these different organs. and it really works every time, basically. So that towards the future, what it means is that the other part of it, and that is where this also why we started working with STEMCELL 7 years ago. We need standardized assay. We need to distribute these protocols. We need to make sure that this a little bit more complex technology, maybe, because it does require that to work in the lab. It is robust, but you need to stick to the protocol. You need to stick to the right reagents. And so the challenge, I think, for Hub and STEMCELL and others in the field is to make sure that we have these protocols available for everyone, that we have the reagents available for everyone, and that we can support the use of it, because we've already, I think, from us and so many others, shown that it is a big change and we can really do something else, but it does rely in a large part on the protocols and on the methods we develop, and also very importantly the quality of the reagents. So I would say the standardization and how do we get everyone to use the protocols that work and to have comparable results, I would say that is what we concentrate on and where we think our challenges are.
If I need to add now new challenges and opportunities to what Rob has already discussed, I think that one of the challenges is that we are putting a lot of effort about the predictive value of the organoids, because we want to make an impact into the patients. But currently, organoids are still experimental. They are not necessarily the first line when we get access to tissue to be able to validate these things. So sometimes we struggle with the amount of material and that we can receive in order to prove this predictive value. But hopefully, with the results that we've presented here about demonstrating their predictability, we will get more chances to implement organoids in new clinical trials where we will have priority in terms of getting the material and generate good quality data. And then I think that one of the important opportunities, especially taking into account that all these technologies started with the possibility of establishing organoids from healthy cells, and all this new development there is in terms of Cas9 technology and developing all these new base editors that prevent much more side effects. I think that there is also a big opportunity in terms of regenerative medicine and being able to repair specific genes in some tissues like liver, for example, and provide an opportunity for these patients to replace cells with a genetic defect with cells with the proper genetic function.
Ryan, do you want to just speak a few words as well in terms of opportunities and challenges?
Sure. I think a lot of them were hit on really nicely by Rob and Sylvia. I think just touching up or adding a touch to that is, I think whereas Rob was speaking of the standardization and really really starting out on an even plane with everybody's organoid cultures. Indeed, this is important. But what it allows us to do is really pick up one of the key benefits of organoids, which they are precision medicine. We're able to detect phenotypes in specific disease patients, and Sylvia highlighted an excellent model in the cystic fibrosis through a very simple assay. They can really pick up the different mutations, look at the different treatment possibilities to that. So by focusing on standardization, we can get into the specific details and really focus on the individual patients as well. I think as far as challenges too, one of our responsibilities is to be extremely diligent in our characterization of the organoid cultures. Single cell sequencing has added a great tool to this, where we can actually look at the composition of the organoids and see what the various assays and culture systems are doing, and with respect to cell population. I also think as we move towards precision medicine and transplantations, we need to focus on what we're scaling up and making sure that the starting population of cells within a dish are going to represent what's going to be going into patients. So again, pushing the field forward and maintaining this pace it's advancing at is incredible, but also making sure we're very diligent as we advance to make sure we're going along the right paths.
Great, thank you so much, Ryan. I believe that's all the time that we have for today. I really would like to obviously thank our presenters, Rob, Sylvia, and Ryan, for their time and excellent presentations. And I would also like to thank all of you for attending and submitting questions and actively participating. So thank you very much, everyone. Have a wonderful day or evening wherever you may be. Goodbye.